In this lesson, we will look at the denaturation of proteins and determine how proteins are denatured. Let's look at the structure of sucrase. The role of sucrase is to help break down sucrose in your body, and without it, we can't get the glucose we need, and our body would not be able to process the sucrose that we consume. If we look at the shape of sucrase, we see it has a very specific shape, and if we looked at any molecule of sucrase, we'd see something that looks very, very similar. However, if something happens and part of this structure is lost, this shape of the molecule is lost, then sucrase can no longer function correctly. We're going to look at what denatures proteins so we know what to avoid, or if we want to denature one, we know how to do it. Another example of an important protein is the potassium channel that helps potassium and the potassium ion pump in and out of your cells. So without that, we would have some major health problems that would, to the extent it would not be compatible with life. So denaturation is what breaks down secondary, tertiary, and quaternary structure. Notice that primary structure was omitted, and this was intentional. The denaturation of a protein does not change the amino acid sequence in the primary structure. What it does is unfold and untwist the secondary, tertiary, and quaternary structures. Because these are held together by the attractive forces similar to intermolecular forces and not held together by bonds. So denaturing will break down forces but not break down bonds. However, this amino acid sequence is useless unless it has the right secondary, tertiary, and quaternary structure. So in order for a protein to function correctly, it must maintain all levels of structure. What level of structure has peptide bonds? So primary structure is where we find peptide bonds. Secondary, tertiary, and quaternary have interactive or attractive forces between them. So we showed the structure of sucrase. Now we have the structure of sucrose. And in order for the sucrose to break down, we have to have sucrase. Without that, there is no reaction. So how do we denature proteins? One way is heat. If we get it above about 50 degrees, the structure of the protein will start to break down. The addition of strong acids or bases, which interfere with those attractive forces between different parts of the amino acid sequence. The addition of ionic compounds, such as salt, so NaCl adding would interfere with those attractive forces. Detergents, heavy metal ions, and agitation. So when you have stains on your clothes, a lot of times they are protein stains, and detergents can help break those down and helpfully remove them from your clothes. What conditions are most likely to result in the denaturation of sucrase? So there's actually two: heating to 45 degrees and adjusting the pH to 6. So the more we heat something, the more likely it is we'll denature it, and changing the pH away from neutral to becoming more acidic is also going to interfere. The exact temperature or pH where a protein or enzyme will start to be denatured will vary depending on the protein and its normal conditions. What type or types of structure are lost in denaturation? The key here is that everything except primary structure is lost. Denaturation does not break the amide bonds in the amino acid backbone.